Hello, good afternoon. Welcome to BSI Conference 2018. My name is John Acquah-Mensah and I'm going to take you through Pica practice in Ghanaian pregnant women, possible role of genes. This will be my outline for the presentation. Pica can be defined as an eating disorder characterized by craving or eating of non-food substances with little or no nutritional value. The condition is common among pregnant women and children. It has been associated with iron deficiency, which is a major cause of anemia. There are two schools of thought. One are with the view that Pica practice leads to anemia, whereas the other are of the view that the dietary irregularities leads to Pica practice. There are various types of Pica. You have geophagia, pagophagia, amylophagia, and trigophagia. These are some of the forms of pica I spoke about a while ago. Pagophagia, the consumption of ice. Geophagia, the consumption of clay. And then trigophagia, the consumption of hair. The sample of baked clay you are seeing in this picture is how the clay is sold in Ghana here for pregnant women to consume. Pica is very prevalent in Africa, and Ghana is not an exception. A study conducted showed that 47% of pregnant women in the Kumasi metropolis practice Pica, whereas in the capital city of Ghana, which is Accra, 48% of pregnant women practice Pica. People practice Pica for various and diverse reasons, some of which include hunger. They eat this baked clay just to satisfy themselves. Others for medicinal purposes. Traditionalists coat their herbal products with this clay so that they can be able to consume. There are possible effects of practicing Pica, some of which include worm infestation. This baked clay consumes heavy microbial load, which will lead to worm infestation. Another one includes increasing the risk of lead and arsenic poison. People chew or eat paint chips, and this contains lead, which is very dangerous to the health of the individual and then to the developing fetus. Pica has been implicated with anemia and remains after iron therapy. It is therefore possible that the malfunction of the iron transport protein could lead to the development of Pica. The temperate gene is a gene that encodes multiprase-2, a serine protease membrane, which is a transmembrane protein, known to be involved in iron metabolism. Ceruloplasmin is also a copper-containing glycoprotein, which helps in iron absorption by transferring. A defect in temperate gene has found to cause iron refractory iron deficiency anemia. And then a defection or a mutation in ceruloplasmin gene has also been linked to low levels of ceruloplasmin, leading to iron deposits in an individual. Bleeding births were found to have high amounts of ceruloplasmin. Now to the problem statement. The history of Pica from the days of Hippocrates till this 21st century has puzzled scientists and researchers due to its unclear etiologies and health implications. Many women practice Pica. They are ashamed of the act, and they don't tell anybody about it, neither do they tell their doctors. This problem has led to it being underdiagnosed, and hence treatment has not been meted out to these people who practice Pica. The main objective of this study was to determine the association of some selected genes to Pica practice in Ghanaian pregnant women, specifically to determine the hematological markers such as the white blood cell count, the mean cell hemoglobin, and then to determine the sickling status in these women. Then finally to determine the presence or the absence of the temperate gene and then the seroplasmin gene in these women using PCR analysis. A case study conducted by Key et al. Revealed that geophagy was the cause of a maternal death in a 31-year-old woman who practiced Pica. They therefore concluded that Pica is a harmful habit and must be handled aggressively. This work seeks to investigate the role of gene in Pica practice. The study locally was at Inwa Medical Center at Emina Bodi, a suburb in Kumasi. Questionnaires were administered to the participants, and then venous blood samples were taken, and further analysis was done to get the various hematological indices. The quality of the DNA was checked using gel electrophoresis, and PCR was also done to confirm the presence or the absence of the various markers in the blood sample. To the results and discussion. This table is the demographic data of the Pica and non-Pica practicing pregnant women. 54% of the participants practiced Pica, whereas 46% did not practice Pica. The age range was between 20 and 39. For the educational level, it was found out that most of the people were having low level education. That is, most of them were below the tertiary level of education. And this agrees with Mohammadi and Mohatabi 2010, which also confirmed that people who practice Pica have low level of education. Now, 22% of the participants also confirmed that they had a family history of Pica. That is, their parents or their mothers practice Pica, and they also have the form of Pica practice in their previous pregnancies. To the hematological indices, there were no significant differences between the hemoglobin level of the Pica practicing and non-Pica practicing pregnant women. The hemoglobin level of the Pica practicing pregnant women were found to be 10.58, whereas that of the non-Pica practicing were found to be 10.77. Using the p-value of 0.005, it was not statistically different. The same applies to the MCV and then the white blood cell count and then the red blood cell count. The only statistically different one was the mean cell hemoglobin concentration, which was found to be, which using a p-value of 0.0232 was found to be higher in Pica practicing pregnant women than Pica non-practicing pregnant women. Now, comparing the sickling status of the Pica practicing pregnant women, once again, we found out that 4 people were negative for the resource factor, whereas all the remaining were positive for the resource factor. Many or most of the people were of genotype AA. 11.11% were of genotype AS, 1.18% were genotype AC, and 1.18% were of genotype CC. For the comparing the hematological indices, most of the hematological indices were not statistically different from the various genotypes, that is AA, AS, and then CC. Now let's look at the gel electrophoresis. The electrogram in figure 5 is showing the bounds of the genomic DNA after isolation, which confirmed that they were good for the PCR rate. Figure 6 shows the electrogram of the bound fragment of the Pica marker, that's the temperate 6 gene. All of the markers were present in all the participants. Figure 7 and Figure 8 is the electrogram showing the Pica markers of the cerebroplasmic gene. That's ACP3334, ACP2324, and then ACP3 and 4. This table shows a summary of the Pica markers, the temperatures gene. All of them were present in all the Pica practicing and non-Pica practicing pregnant women. This table shows the summary of the cerebroplasmic gene genetic markers. The sum total reduced to 52 and 53 because this particular study was carried out a year after the temperatures gene was carried out. So we have a reduction in the number of DNA which were which qualified or which were good to be used for the PCR rate due to the storage and then power fluctuations. It causes some of the DNA to degrade, so the number reduced. Now looking at ACP3 and 4, ACP13 and 14, ACP33 and 34, all of them were present in both Pica practicing and non-Pica practicing pregnant women. However, their prevalence were higher in Pica practicing than non-Pica practicing pregnant women. But for ACP 11 and 12, 23 and 24, 25 and 26, all of them were absent in both Pica practicing pregnant women and non-Pica practicing pregnant women. We cannot conclude on the fact that these genes were present and these genes were not present to be used as a basis of it being linked to Pica. However, further studies can be done on checking whether there is a mutation in those genes which were present and then those which were not. In conclusion, Pica is common among pregnant women attending in one medical hospital. Pica has considerable effect on their hemoglobin levels, but little significant difference in other indices used. PCR confirmed Pica and temperatures genes present in other subjects. 3 cerebroplasmic markers, that is ACP 1112, 2324, and 2526, were completely absent, whilst another 3, ACP 34, 13 and 14, 33 and 34, were present in Pica practicing and non-Pica practicing pregnant women, but they were higher in the Pica practicing pregnant women compared to non-Pica practicing pregnant women. Other primers should be used in further research studies. For the cerebroplasmic gene, we have about 18 primers, but we use only 6. So other primers can also be used for further studies. Now, molecular analysis should be done or can be done in that the mutation can be checked using certain blotting and then restrictive fragment length polymorphism, that's RFLP, in checking whether there is a mutation or not. These are my co-authors, and here are my references. Thank you very much for your time.